Hello, in this video we will discuss about the CRISPR-Cas9 system. What is the CRISPR-Cas9? And we will also learn about the biotechnological working through CRISPR-Cas9. So the CRISPR-Cas9 is containing gene which that is contain a palindromic sequence, which that is here is the new spacer can be bind with the repeat. So the palindromic repeat is this is the repeat palindromic we will also learn, and this is the plasmid of the bacteria, and this is the DNA in the form of chromosome, you can say. And this is the E. coli bacteria. So this is the bacterial genome. This is not bacterial genome, this is the bacteriophage genome. So remember, this is wrong thing. So in this way, here is the promoter, which that is a use for the promotion of the Cas9 gene activation to produce. We will also learn. So the CRISPR-Cas9 is the clustered regularly interspaced short palindromic repeats. Short palindromic repeats is a natural part of the genome. So remember, and found in bacteria and archaea. So CRISPR means the grouping of short palindromic sequences separated from each other on regular basis that isolates DNA sequences recognized foreign spacer. So whenever the bacteriophage will be phage to invade the viral DNA, which that will be bound as a new spacer in the plasmid. After the binding with the new spacer in the plasmid, so the palindromic sequence repeat is also present here. So after this, the new spacer when bind, in this way the new spacer will be also replicate but also transcribe, and the transcription will lead to produce the mature CRISPR RNA, while this is the Cas gene will produce through promoter to activation to produce a Cas9 complex. So in this way, here is the plasmid extra DNA in the bacteria, and this is the palindromic repeat. What is this? This is here you can understand the palindromic repeat mean the cytosine guanine toward the 3' to 5', while the guanine with cytosine is here, which that become a hairpin like structure when bind together after the transcribing. So this is between the repeat palindromic repeat, the spacer are present. This spacer can be come from the bacteriophage DNA when bind through a ligase enzyme. So remember, this is a structure of basically is the structure of uh, uh, a cruciform hairpin DNA with a palindromic sequence. So in this way, after this is the transcription to produce the mature CRISPR RNA. This mature CRISPR RNA means the cluster regularly interspaced short palindromic repeats, which that is that RNA in the form, and in this way it will be bind with the Cas9 complex, and in this way the CRISPR RNA will bind here, and recognition site will allow for the restriction of the endonuclease enzyme use to viral genome cutting. After the viral genome cutting to degradation of the viral genome, and in this way the viral disease and the viral replication of the genome will be impossible. So that is the major reason this is the defense system, adaptive defense system, because the new spacer will remain in the plasmid, and this is the basically the defense system, adaptive defense system are present in the CRISPR-Cas9 system. So on the other hand, here let's begin to understand the genome editing with Cas9 nuclease enzyme. So the nuclease mean it can be cut, restriction enzyme, but remember this is the Cas complex, Cas9 complex, and this is the DNA, the genomic DNA. And in this way the target sequence are present here, which that will be Cas9 will target and bind this protein, and the single guide RNA, which that is chimeric guide RNA, contain a CRISPR RNA plus track RNA, which that will track the site of the specific site for the restriction. And here the pathogen associated molecular sequence so will cause the pathogenicity. So that will be, here is a single guide RNA will guide and bind with the specific site of the PAM sequence and the target sequence. In this way, this is the target. The single guide RNA will target this target sequence. Pathogen associated molecular sequence, basically the pathogen associated molecular sequence will be cut, and in this way the CRISPR RNA will bind by this and to cut, and the two situation can be occur, the NHEJ non-homologous end joining and HDR the homology direct repair, which that involve the Q70, 80 ligase and the ATM and ATR and RAD 55. 51. So in this way indel mutation can be occur. Indel mutation mean not donor DNA involved, but it will be repair. But in this case the target genome editing through donor DNA through a HDR the homology direct repair. So in this way you can understand as well, this is the direct repair, while the non-homologous end joining, basically the non-homologous end joining not repair but indel mutation. In this way the excision of the specific genome for prevention of the disease and other things. So remember the genome editing by double nicking also occur. What is the meaning of this? This meaning the target sequence 1 and target sequence 2 involve, and this target sequence 1 and 2 involve to produce to restrict and cutting the domain, and in this way the HDR homology direct repair, and in this way the ATM ATR RAD5 prime, which that will lead to restriction, and in this way you can understand this will lead to repair through donor DNA. The donor DNA is providing through the DNA interest, which that will be bind and the targeting sequence here, and this will lead to targeting sequence will bind with the donor DNA. So remember the donor DNA will bind, here is the ATM ATR and RAD 5 prime. So after this the target sequence, which that will a donor DNA basically will bind. After this is the 3 prime to 5 prime direction, and you can understand the 3 prime to 5 prime direction restriction different origin will lead to ligation through a donor DNA. So this is the genome editing by double nicking. While on the other hand, the third one is the visualization and localization. The visualization mean we can visualize, and we can say that is basically, we can say that is the GFP is involved. GFP will produce color, means means the restriction will be complete, and the cutting domain is here HNH and RUVC, which that is used for the cutting of the domain, the cutting of the domain. So after the cutting of the domain here you can understand, so the transcription regulation, which that will be occur. The transcription regulation is the fourth part. In this way that was the GFP will indicate that the cutting domain will complete, and the cutting the restriction of the unrequired genome will be cut. So on the other hand, the transcription regulation means the RUV and HNH, which that will be cut. After this, the cutting, the effector, the target gene, which that will be effector will inhibit to produce the red light, and activator will produce the green color. And in this way you can understand the transcription regulation, which that means the transcription and regulation will be changed, lead to effector. That effector will produce color, the red and the green. Green is the activator. So remember these things. So red is used for the replication, so the inhibition of the replication, not basically replication, while the activator is show the green color, which that replication will inhibit and activator will be activate to effector, lead to replication inhibition. So in this way the replication of the unrequired genome will be inhibited by this mechanism. So this is the CRISPR-Cas9 system, which that is used hugely in the biotechnology field for cure the disease and synthesis of drugs on the basis of genetics, as well as other properties which that I have mentioned in the form of genome editing. So the basically the CRISPR-Cas9 system is the genetically editing system. So thanks for watching.